Hi, my name is Gretchen Pritchard from the University of Washington, and I am excited to tell you about our new paper entitled BCL6 is required for the development of functionally responsive IgM GC Tfh independent memory B cells. In this study, we are looking at the developmental history of specific memory B cell subsets. During T-cell dependent settings, after initial interactions with CD4 T cells at the TB border, some memory cells form in a germinal center independent manner, whereas others experience the GC on their way to memory formation. Within the GC, B cells interact with GC T follicular helper cells, and the resulting GC-derived memory B cells have undergone class switching and somatic hypermutation and have phenotypic markers associated with GC history, such as CD73, CD80, and PDL2. In the Pepper lab, we used B-cell tetramers to study antigen-specific B-cell responses that are induced due to infection or immunization. By using magnetic enrichment strategies, we are able to study rare B-cell populations. And importantly, we use a decoy tetramer that contains all of the aspects of our tetramer, minus the antigen, with an additional fluorophore conjugated so that we can identify the polyclonal endogenous B cells specific for our antigen of interest. In these studies, we primarily looked at responses to a protein called MSP1 from Plasmodium, the parasite that causes malaria. Several years ago, a previous graduate student, Akshay Krishnamurti, found that after Plasmodium infection, the IgD B cells looked like naive cells. However, the IgM memory B cells were phenotypically similar to the class switch memory B cells and had BCRs that had been mutated. This made us wonder whether these IgM memory B cells were GC dependent despite having retained their IgM BCR. BCL6 is a transcription factor that is expressed in both B cells and T cells in the germinal center and is in fact required for GC development. So to differentiate the effects of intrinsic BCL6 expression from the presence of the germinal center, we devised a strategy whereby we generated conditional knockout mice so that we deleted BCL6 from either the B cells or the T cells, and in both of these systems, the germinal center is not able to form. We found that when BCL6 is absent from the B cells, neither IgM nor class switch memory B cell subsets were generated. However, when we deleted BCL6 from the T cell compartment, we saw that the class switch population was reduced, yet the IgM memory B cell population was intact and had even undergone mutation of the BCR. Looking a little further, when we depleted CD4 T cells in vivo, we saw that BCL6 was not expressed in the B cells if the T cells were not present during early activation. However, in mice in which the T cells are present but can't express BCL6 and thus are unable to form germinal center Tfh cells, the B cells are still able to express BCL6. We have added to the knowledge in the field about how some memory B cells are formed, specifically the IgM memory B cells that are phenotypically similar to class switch memory B cells. Both populations require intrinsic BCL6 expression that is dependent on the presence of CD4 T cells. However, the IgM memory B cells do not need to go through a germinal center on their way to becoming a memory B cell. These studies help us to better understand the developmental requirements of functional memory B cell populations. And with that, I'd like to thank our collaborators and thank you for watching. I hope you check out our paper.